Okay, so if you look at the handout then on plant structure, which looks like this. This is just, very, very basic. This top diagram is background information, so you can write that it's sort of just general knowledge. You're not going to get a diagram like this and be asked to label it. But obviously the bit that is above the ground is the shoot. So if you want to draw a dotted line and write shoot and root. So if this is a tomato plant, so there's tomatoes are actually fruits, okay? You maybe think of them as vegetables, they are actually fruits. So that's like the tomato. Inside it, there'll be seeds. And that's how the plant reproduces. It makes seeds. An animal comes along, eats the nice fruit. The fruit gets digested. The seeds don't get digested. They pass through the animal and get deposited in the feces wherever, hopefully somewhere further away. And that's how the seeds get dispersed. And then they germinate and they grow into a new tomato plant. So obviously we've got leaves there. And in terms of, there are words like the apical bud. And that's already labelled there. The apical bud. Have you come across the term apex in triangles and things like that? So apical means to do with the apex of something, so like the tip. So that's the apical bud. And then these side buds are called lateral. So lateral means like sideways. So lateral, we've also got lateral roots. And that's labelled as well. The main root is called the primary root. At the bottom there, we've got root hair cells with the little root hairs coming out. And then we've got the root cap and the root tip there at the bottom. Where does water get into the plant? Zara, where do you think water gets into the plant? In through the leaves, the roots. The what? Where, Penny? The roots. Okay, what cells? Root hair cells, okay. So water gets in through the roots, and then there is this transport system here. Okay, and so we've got this transport tissue here, which is in the middle of the roots, and then it sort of splits up and goes into the stem, and you see it goes up through the stem, and there are branches of it coming out to the lateral shoots and then out to the leaves, and you can see that happening throughout the whole plant there. All right, so that tissue is the equivalent of your arteries and veins. Now, it's not at all like the same structure, but it does the same job. It transports water, it transports sugars and other nutrients. And your blood vessels are part of your vascular system, and so we use the same term in plants. This is part of the plant's vascular tissue, and you can see there that it's labelled like that on your diagram. Okay, so that tissue there is the vascular tissue. And what are we talking about? We're talking about xylem vessels and phloem vessels that you've been reading about. So if we go down here to the roots, and if you were just to take a knife and cut through there, and then just take a razor blade and cut a very small slice of that and put it under the microscope, you are going to see, we'll see in the root, the vascular tissue is in the middle, but it sort of branches off. And actually what happens is we get little bundles of vascular tissue in the stem. All right, so this kind of set up here. Now, it's not going to look, this is what's called like a block diagram, and you looked at block diagrams. We've drawn block diagrams before where you don't draw the individual cells, you just draw the areas of the different tissues. And so you need to know this is a transverse section, that means like a cross section, through a dicotyledonous root. That's what your specification talks about. It's the only kind of plant you need to know about, a dicotyledonous plant. It's a root, hold on, Lee. Take this phone call here. So, what labels are we going to put on this? What are we looking at here? What is the outer layer called? From what you've been looking at in your notes, what's the top label there, the very outer layer called? Epidermis, okay? So let's add some labels here. What's the next layer then, moving inwards? Cortex. And then inside, we have this layer, or this whole area, which is called the stele. So we're going to put another label at the other side, and this is STELE. That whole structure and that the stele contains the vascular tissue. So which is xylem and which is phloem? So which one is in the middle? Xylem. And what does xylem carry? Water, okay? And then on my diagram, it's the yellow pockets there are the phloem vessels, and they take the sugars that are made in photosynthesis. And then there's a circle of endodermis. Now in biology, epi means on the outside, endo means inside. So epidermis is the layer, the lining tissue on the outside. Endodermis is a lining layer of tissue, but it's not on the outside. It's inside. Okay. So if we look at this photograph here, then we can put some labels. So we can't see the outside of the stem, so we can't see the epidermis, but we know, I'm going to get you to label that as the stele. So, what's the top line pointing to? Amy, what do you think? Yeah, endodermis. Jacob, what's the next one? Xylem. And then the last one is the phloem. So you'll see there that the xylem vessels are massive. They are really, really big. Now, one of the skills that you need to develop, and certainly if you're doing the practical exam, I mean, you all need to do this because next year you'll have to do this anyway. But a block diagram, you're going to draw a block diagram. So in the practical exam, you might get a photograph like that, and it might say, this is a transverse section through part of a root. Draw a block diagram and label at least 4 structures, all right? So a block diagram, you're not drawing the individual cells, you're drawing the blocks of tissue. So tissues are similar cells, so xylem tissue, phloem tissue, and so on. So it has to be drawn, it has to look like the photograph. So, it's drawn to the same size, so roughly, you want your steel, you want your, I'm not very good at drawing here, but that's, okay, so it should be sort of the same shape and the same size, so we want to make that the endodermis. No, that's not brilliant, but you get the idea. It's quite hard to draw on the iPad. And then obviously then the xylem, and if we look at this is the xylem. So you need to have that looking something like that. It's not brilliant, but it's not bad. Now obviously you're using a ruler, and then phloem. Okay, it's only 3 tissues. One other label. Well, what about all these other cells that are around here? What are they part of? Cortex. So, cortex. We should also put the cortex in the top as well. Now you can sort of bracket those together and write steel. Let me see. At this diagram, this is a typical diagram of what it looks like if you were to cut through a stem. So this is in your handout that we were labelling there. So again, we've got the epidermis on the outside. Okay. Now, in the steel, which vascular tissue was in the centre? Xylem or phloem? Xylem. So that means on my diagram, the pink one is going to be xylem. It's towards the middle. And then the blue one on my diagram is the phloem. On the outer edge, just we've got the cortex. And in the middle, we've got, this is called the pith. And one of these things here is called a vascular bundle. Basically, that diagram is sort of that bit there. Okay? And if you look at this, we've got the epidermis at the top here. What's this stuff here? Well, it's the cortex, isn't it? So we'd be taking, this would be down there. All right? So you need, so if we look at this diagram on the next page then, this is taken from like the top of that diagram you have. If you look at that, so you can see each of those is a vascular bundle. Now you don't need to draw that. What we'll do is we'll add some labels just, all right? So I suppose we should say this is a transverse section of the stem. So this outer layer is the epidermis. In your diagram, you're just going to put a label there, cortex. I would label that as a vascular bundle. Now, if we look at this in more detail, this is actually the vascular, well, I suppose I really should have done that as a vascular bundle, don't worry. The large vessels are the xylem vessels, and they're the ones. Right, so we said the xylem vessels are the large hollow ones in there. Anyway, they're the ones that are close to the centre. These ones are phloem. In this diagram, we have this is actually a big bunch of fibre, which really gives a lot of support to the plant. Okay, underneath it, we have another photograph of a cross-section through a stem, and this one's showing a vascular bundle. So if I sort of go over the outline of the vascular bundle, do it in black. That's the vascular bundle there. What do you notice about it? Where's the xylem? The top? Yeah. So this is sort of, this photograph for things like that. So in this case, we're going to draw a block diagram. So there's your vascular bundle. That's all you need to draw for the shape. And then obviously your labels are xylem, phloem, vascular bundle. These are fibers. Cortex, because it's towards the outside. And this bit here is the pith, the cells that are towards the centre of the stem. Okay. I'll get you a few slides here. Okay, so if you go back to the handout that you got, this diagram here is basically showing you a cross-section through a leaf. So as I said, the xylem vessels are the bits at the top, phloem underneath, but there'll be little branches coming off that. So if you think of the structure of a leaf, so there's your stem. Xylem vessels at the middle. Okay. I'll draw the phloem vessels in green there towards the outside. It ends up at the bottom. Okay, so you're getting these veins, okay, which have got xylem vessels and phloem vessels. When you cut through that, if you were to cut through there, you're going to cut through a number of veins, and that's why you see these here, okay, because you're cutting through a number of these veins, okay, they're vascular bundles, to give them their proper name. So the xylem's always on the top, phloem on the bottom, so xylem would be there, phloem underneath, and the same is going to be true in the leaf here. All right, does that make sense then? Okay. So xylem vessels are large hollow tubes. Xylem vessels, when the plant was growing, xylem vessels would have started off as big long cells joined end to end. Now, as the plant developed, a substance called lignin is deposited on the walls of these vessels. Now, lignin, have you heard of lignin? Anything to do with that? No? Lignite? No? Okay, so lignin is a chemical. It's really a woody substance. Plants, like trees, have got lots of lignin in them because the tree trunk is nearly all xylem vessels. Okay, so lignin is a waterproof chemical, and so when these cells were being made and developed, the lignin was deposited in the walls of the cells. Okay, so the side walls of these cells became lignified. Now, I said it's waterproof. If I covered you in a plastic bag, what's going to happen to you? What's going to happen to you? If I put you in a plastic bag and sealed it, you die. And that's what happens to these cells. Okay? They die. So mature xylem vessels are dead. Okay? Because they've been covered in lignin and they die. And so a mature xylem vessel basically is hollow. It doesn't, the cytoplasm is removed. It's a hollow series of xylem vessels, okay? So we don't really call them cells anymore. We call them xylem vessels. And the walls are thick. They're lignified. And if you were to take a cross-section through there, they're not round. They're sort of more that kind of shape there. The big, thick lignin wall around them. Okay. Lignin is really, really strong. Okay, so xylem vessels will give the plant support. And as I mentioned, a tree has got just like millions of xylem vessels. That's what makes up the tree trunk. And when you if somebody cuts through a tree trunk and you see the rings, those are rings of xylem vessels. And the reason why they look like rings is because in the early part of the growing season, the rings are biggest. And then as the season goes on, the rings get smaller. And then the next spring, you get another bigger ring of bigger vessels. And so it appears as like concentric rings. And that's how you can count the age of the tree. But anyway, getting back, like plants outside there, like a rose bush or any kind of ordinary sort of small plant that's not a woody plant, basically it gets its support from the xylem vessels because lignin is such a strong chemical. So as the cells are developing, They die and they become hollow, so the cytoplasm is removed. So basically they become hollow, and also the end walls are broken down as well. And so instead of having a stack of cells, it actually looks, those get broken down. So it's almost like just a straw, basically. Now, obviously water has to move out, and so there are sort of pits in the walls to allow water to move out. So there will be sort of little tiny gaps, and that's much too wide there. So there will be small pits at the side there to allow water to move out of the xylem vessels. All right, so they only transport water and the minerals that are dissolved in the water, so nitrates and phosphates and so on, that will be dissolved in the water. They will transport the water and those dissolved minerals up from the roots, up through the stem and out to the leaves and so on. So they're lignified. As the cell matures, you get extra layers of lignin called secondary thickening deposited. And you can see there in your notes that sometimes these are rings, like those ones there. Sometimes they're spirals. Okay. But as they get a wee bit older, then there's more and more deposited. So it looks like that, which is called reticulate. And then towards the end, it looks pitted like that. So in the early stages, it's spiral, and that's good because you can stretch a spiral. So as the plant is still growing, the spiral basically can get pulled, and the vessels can get lengthened that way. All right. So the very first xylem at the it's called protoxylem. So a prototype is like a model you make at the beginning to see whether your racing car is going to work or not. So protoxylem is early xylem. And so we've got the spiral or the rings there, the annular thickening. And then in the mature parts of the plant, it'll look more like this, where there's more and more lignin like that. All right. And the little pits, the little holes there allow water to move out, sideways out to the like side shoots, lateral shoots, and so on. Okay. Right. Phloem vessels, on the other hand, are different. Phloem vessels again are stacked end to end like this, and they have cellulose cell walls. So there's no lignin. They are living cells, but they are specialized. So each we call these vessels here the sieve tube elements. And this is where the sugars are moved through here. Now, these are pretty hollow. They don't really have very much in them. They might have a little bit of sort of cytoplasm at the end there, but basically the sugar solution is just moving up and down through the middle of these cells. And so there's not really room for all the mitochondria, rough endoplasmic reticulum and stuff like that. And so every sieve tube element has got a companion cell beside it, and that contains all the sort of the normal organelles and all the sort of the stuff. It has the nucleus and so on. So the companion cells beside it with the normal cellular contents. These are called sieve tubes because if you look at the end walls, so the end walls, this bit here, actually they have become pitted. That means there's holes in them. And if you were to look down at one of these, it would look like that. It's got lots of holes in it to allow the sugars to move through there. So it's hasn't been broken down like in the xylem vessels, but it looks like that. Now, what do you use in a kitchen that has lots of holes in it? Sieve. So that's called a sieve plate. So that bit there is a sieve plate. It would look like that, has lots of holes in it to allow the sap, really, the phloem sap, which is lots of sugars, to move from cell to cell. Okay? So this is the sieve tube element. Let me point here. These are the sieve tube elements here. There's one cell, and there's a sieve plate there. Each will have a companion cell, which has got the nucleus and all the normal things here. These are very metabolically active. They will produce a lot of energy because sometimes active transport is needed to load up the sugars into the phloem vessels. Okay, I think we will leave it there. So look, what I want you to do for homework is to read through what we did today. But also you're doing a test next week on that chapter on surface area to volume ratio, transport systems, gas exchange surfaces, breathing. All right? If we look at this, we just look at this on the screen here. Again, it might say this is a photograph of a stele of a dicotyledonous plant. Label, we'll say 1, 2, and 3. So what's 1 going to be? It's not. You don't have a copy of this, all right? I'm just doing it on the board. What's 1? Louisa? Endodermis. Caris 2. Phloem, and that means 3 is the xylem. All right? We go back. If we go back then to your handout. Just a reminder, we would have looked at this diagram in year 11 when we looked at cross-section through a leaf, and then obviously around Christmas time when we looked at the structure of the leaf and the tissues there. So you're aware that you've got the upper epidermis, the palisade mesophyll, the spongy mesophyll. What I'm showing you here, this is xylem and phloem vessels. Now, if you think about how it works, if you have a stem, all right, and you've got vascular bundle there, and if you've got the xylem vessels towards the middle, so the xylem vessels, well, I'll draw them in, colour them in blue, all right. Xylem vessels are towards the middle there. If you take a little side branch off that, your vascular bundle is going to sort of continue out there. Those vessels are going to continue. So whenever you see them, that's going to be at the top, and the phloem, which are normally on the outside, are going to be at the bottom. Logically, if you carry that on out. So when you look at this diagram, you know that we've said the xylem, which are nearest the middle, end up at the top. And you know that the xylem vessels are the larger vessels. So there, the top ones there, that I've just coloured in blue, that's xylem, and underneath then we've got phloem. Right, we're not getting them. Right, well, don't worry, we'll come back to this.